I think this has been a very enjoyable day, hearing the different opinions. And sometimes I think that it's helpful to actually try to understand the biochemistry and the science behind it. And so this talk is aimed at trying to understand the science, and I apologize if we get into a little bit of chemistry here. Now, we talked this morning about how if you feed an animal fructose, they become leptin resistant, and that this leads them to eat more than they normally should, and they gain weight. The gaining of the weight is primarily from the leptin resistance and the decrease in fat oxidation that occurs in the liver. So the reason sugar increases weight is because it encourages you to eat more. So if you do isocaloric trials, as you just heard, you'll never see weight gain, because when you do isocaloric trials, you're restricting the human from being able to eat what they want to eat. So all isocaloric trials will always show no difference in weight unless there's a very significant decrease in metabolism. And the decrease in metabolism is really quite minor. It leads to, you can't pick it up in a 4-week study. So all these studies that say sugar doesn't cause weight gain in isocaloric trials is expected, because the way sugar causes weight gain is through inducing leptin resistance and blocking fat oxidation. But what you did hear today is that isocaloric studies do show that fructose increases lipids and has effects independent of weight gain. And this is a recurrent theme. And in this study, which I showed this morning, if you take animals and you give them isocaloric fructose or sugar versus starch or glucose, you can show a whole range of metabolic effects. In fact, as I told you this morning, you can even calorically restrict animals and induce not only fatty liver, not only insulin resistance, but actually diabetes. So the key question that we should be asking is, how is fructose doing this? Why should we pick this one nutrient? And why is it causing diabetes and insulin resistance and elevated triglycerides, as you heard in even isocaloric studies? And it would even show it in restricted calorie studies, I predict. And why? What's the mechanism? So in order to get there, we have to understand how fructose is metabolized, because it's something specific about fructose. So here's an amazing bit of knowledge, okay? Fructose is the only sugar that depletes energy in the cell before it makes energy. All nutrients are used to make energy. We produce the energy in little organelles inside our cell called mitochondria. And these mitochondria produce energy in the form of ATP that we use to drive all of our biologic processes. When you take fructose, the cell consumes ATP in its metabolism of fructose. And it consumes so much ATP, so much energy, that the energy levels fall to an alarm state, a mayday state in the cell. It's really a form of alarm. It's a mayday. And the energy levels fall so low that there is inflammation, oxidative stress, and even an arrest in protein synthesis. A study done 20 years ago showed that they took people, this was a human study published in the European Journal of Clinical Investigation, in which they were operating on gallbladders. They were removing gallbladders from patients with gallstones. And during the surgery, they got IRB approval to either inject intravenous glucose or intravenous fructose and take a snippet of the liver during the surgery. And what they showed is that when they gave fructose, there was a loss of ATP, a loss of energy in the liver, and that was associated with oxidative stress and a complete arrest of protein synthesis. In recent years, they've used NMR and all these kind of fancy imaging, and you can measure ATP levels following the injection of fructose. And I've been an author on some of those studies. And ATP levels plummet with intravenous fructose, but guess what? They plummet with oral soft drinks. With a 12-ounce or 20-ounce soft drink, you can measure the fall in ATP in a human. So it's happening all the time. This energy depletion turns out to be the key. So what happens is fructose, here's the chemistry, fructose gets metabolized by an enzyme called fructokinase. And that phosphorylates fructose to fructose 1-phosphate. And in that process, ATP gets consumed, and fructose depletes ATP. And we're going to talk about this sidearm of this pathway. And then fructose goes on to make triglycerides, which is fat, glucose, and glycogen, which is multiple glucose molecules. And so this has been known for some time. Now, glucose gets phosphorylated, and a lot of proteins and other things get phosphorylated, but classically sugars. And when glucose gets phosphorylated, there is a feedback system that prevents ATP depletion. But when fructose gets into a cell, there's no negative feedback. Instead, the cell will consume all the ATP, it will metabolize the fructose as fast as it can. The ATP levels plummet. It's like a runaway train. There is like nothing stopping the reaction. As little as 1 millimolar fructose will cause ATP depletion in liver cells, in tubular cells. We've shown it in intestinal epithelial cells, in cells where this enzyme is present. So we actually made mice that don't make fructose, that don't make fructokinase. And actually, there's a human disease called essential fructosuria where the people lack this enzyme. And by the way, they do not get type 2 diabetes. They do not get obesity. And in fact, there's never been a case reported with type 2 diabetes or obesity if you have essential fructosuria. And they live a normal life. Anyway, when you block fructokinase, you block this pathway, and of course you block the principal way fructose is metabolized. Fructose can be metabolized other ways, but when you do that, you get this very expected finding. So if you feed fructose to animals, I told you they get fat, they get insulin resistant, they get fatty liver. And here you can see the fatty liver in a fructose-fed wild-type mouse. And this is all fat globules. This is a liver tissue. And the fructokinase knockout they don't become insulin resistant, they don't get fat, they don't get fatty liver. We could show that this enzyme is obviously important. But this was expected, right? Because you're blocking fructose metabolism. It's not earth science, If fructose causes fatty liver and I block the principal way it's metabolized, I'm going to have a benefit. But here's the key thing. There's actually 2 fructokinases. There's one that's called fructokinase C, which depletes ATP. And then there's one called fructokinase A, which is also in the liver, and it does not consume ATP very well because it's a much slower reaction. So now I can do a study where I can knock out one fructokinase versus the other. And if I knock out fructokinase A, basically what I've done is this just shows that fructokinase A does not cause much ATP consumption, fructokinase C does. So if I knock out fructokinase A, basically all the fructose goes through the fructokinase C pathway because now there's no fructokinase A around. And so when we did that, we made mice that don't have fructokinase A. So now fructokinase C is still active, so you have this big sidearm still happening. And now the animals are all fed the same amount of fructose, and the fructokinase A knockout become much fatter. They have much fatter livers. They become much more insulin resistant. They have more, basically all the metabolic syndrome is worsened. They have more increase in body weight, epididymal fat, hyperinsulinemia, elevated liver triglycerides. And so this gave us the clue. It's the ATP depletion. It's the energy depletion that is the problem. It's that quick consumption, quick ATP burning, the big rush, and then the big fall. And it's the fall that tries to tell the cell, hey, look, I don't have enough energy inside the cell. I need to hold on to fat. I need to make fat. I need to save myself. That's teleologic, but I'm not sure that the cell really thinks that. But the fact is that ATP depletion actually seems to be key. Now, how did we know that? Well, what we started to do is we then started knocking out different pathways from this pathway from fructose to uric acid, for example. So when ATP gets consumed, fructose activates this enzyme called AMP deaminase. And we have some beautiful papers now coming out or published, some have been published, showing that AMP deaminase drives fat accumulation. And AMP deaminase turns out to be the inhibitor for AMP kinase, which is a very important enzyme. Actually, the metformin, the drug we use to treat diabetes, stimulates AMP kinase, and that's the way it works. But because AMP kinase is knocked down in diabetes. But guess what knocks it down? AMP deaminase knocks it down, and guess what activates AMP deaminase? Fructose. And what happens if you block AMP deaminase? So this is where we take liver cells and we feed them fructose. So when you feed a cell fructose, the fat goes up in the cell. But if we silence this enzyme, AMP deaminase, we block the increase in triglycerides. Well, that's interesting because the fructose still can be metabolized all the way down. But there was this mystery that was noted 20 years ago, and the mystery was that if you label the fructose molecule, very little of it, only 3% of it gets in the triglycerides. Yet you see a big rise in triglycerides. So the soft drink industry has argued that fructose doesn't raise lipids because when you label the fructose molecule, only 3% of it ends up in triglycerides. But when you give it to people, you see this big rise in triglycerides. And the reason is because the triglyceride rise isn't coming from the fructose molecule. It's coming from the sidearm, most of it. And the sidearm is using glucose and other ways to make fat. And that's probably why you need glucose and fructose together. You need to have the fuel, but the fructose is the fire that activates the machinery. But then other foods are being used to actually drive the obesity, and we can show this in animals. So the AMP deaminase produces a compound called IMP. Sorry about the biochemistry, but the IMP is then broken down to a substance called uric acid, and uric acid can be blocked by this enzyme called, or is metabolized by this enzyme called xanthine oxidase, and you can block xanthine oxidase. And here's when you give fructose, uric acid goes up in the blood. It always goes up in the blood. The paper by Stephen Piper that says it doesn't go up in the blood, you know what they did? They gave fructose during the day, and then they stopped at 8 p.m., and then they had them fast overnight, and then they measured the uric acid. Well, I could show that glucose does not stimulate insulin by doing that same experiment. I'll give glucose during the day, I'll fast them overnight, and I'll measure the insulin. Okay? So, but if we block xanthine oxidase, I can block the formation of uric acid. And what's interesting is when you give fructose to an animal, in the mouse, you have to actually give a lot of fructose to raise the uric acid. And when you do, when you lower the uric acid, you lower the blood pressure, and you improve the metabolic syndrome. It's amazing. But we and our other groups now have shown that if you give allopurinol, this drug that is out there, you can actually block the metabolic syndrome in animals. But the trick in animals is you have to give enough allopurinol to lower intracellular uric acid, and it turns out to be at doses higher than we currently give to humans. So we're giving doses that block serum uric acid, but to actually block this reaction inside the cell, you have to give higher doses than we currently give people. And when you do that, you can knock down and prevent, you can reduce the development of fatty liver from sugar, and you can reduce the metabolic syndrome. And then we went on and we put uric acid on liver cells, and we showed that uric acid actually stimulates fat in the liver cell. This is a stain where you use oil red O, and so red means fat. You put uric acid on a liver cell, suddenly you can see a stepwise increase in fat. And when we went on and studied it, we found the mechanism. The mechanism is oxidative stress. And you saw it in the beautiful slides by Rob, all the ROS, which means oxidative stress. When you put fructose on a cell, you get an immediate burst in oxidative stress, and it's in the mitochondria, the place where the energy is produced. If you put uric acid on the cell, you see the same reaction. And if you give a drug that blocks the uric acid formation from fructose, you actually reduce or inhibit that oxidative stress. In turn, we went on and showed that the way the oxidative stress works is the oxidative stress blocks an enzyme. More biochemistry, sorry about this, okay? It blocks an enzyme in the Krebs cycle. That causes citrate to accumulate. It moves into the cytoplasm and increases fat synthesis. It blocks another enzyme in the fatty acid oxidation cycle, the beta fatty acid cycle. These two enzymes are known to be oxidative stress dependent, and that blocks fat oxidation. Bingo, you've got it. Here's how it works. You give sugar, and you get this biochemical reaction. Energy falls, you get this uric acid formation. It actually enters the mitochondria. You get this oxidative burst. Suddenly, you're making less energy, and the cell goes into a mode to make fat. And you know what? The same reaction occurs when we study other animals that when they turn on and want to gain fat, they activate the same general pathway. This actually looks like a switch, a system for stimulating fat. So what I've just told you is that fructose is the only common nutrient that causes energy depletion before it stimulates energy production. Studies suggest it's the energy depletion that is largely responsible for fructose effects. And these studies actually suggest that not only fructose, but that uric acid may be a true risk factor for metabolic syndrome. So that's kind of interesting because we think of uric acid, we think of gout. Gout is a huge problem in the Maori. Gout is a huge problem in the metabolic syndrome. Gout has been associated with sugar intake. The overwhelming evidence shows that soft drinks increase uric acid and increase the risk for gout. But what's interesting is patients with gout frequently are obese. They frequently have metabolic syndrome. They frequently are hypertensive. They've got kidney disease. They have cardiovascular disease. And we've always thought of the uric acid as being secondary to being fat or secondary to having insulin resistance. But look at this. Here's a paper. They took 433 lean men. They were young. Their BMI is all less than 25. And the ones who had a uric acid of 5.5 had a 16-pound weight gain over the next 5 years, I think. And there was a 4-pound weight gain in those with a uric acid less than 4. There was the only risk factor was just the uric acid level. Uric acid level independently predicted obesity. It was quite a significant effect. And uric acid also predicts diabetes. Now this is from my paper where I kind of reviewed the literature. And look at all the studies that found uric acid be an independent risk factor for predicting diabetes, 23 out of 24. This means that if you have a uric acid, even if you're thin and otherwise completely normal, you have a uric acid, you're at increased risk for developing diabetes, you're at increased risk for developing obesity, you're at increased risk for developing hypertension. Look at this, yes. They're all independent, 18 of 19 studies. And as some of you know, we have shown experimentally that raising uric acid increases blood pressure. And we've also done clinical trials. We published in the JAMA that lowering uric acid lowers blood pressure in obese adolescents. So it looks like uric acid could really be a true cardiovascular risk factor. It may be the mechanism by which soft drinks are driving hypertension and kidney disease and cardiovascular disease. There are many papers that are still coming out that will support this general hypothesis. So then why? Why are we so sensitive to sugar? Why is it that this is actually driving this whole process? And I'm going to go back 50 years ago to Jim Neal, who was an anthropologist at the University of Michigan, who was one of the first people to do medical genetic epidemiologic studies. He was really a leader in the field of medicine. And he wrote a paper where he said, you know what? I think the reason for this rise in diabetes and obesity today is because there were periods in our past where there was food shortage or there was a famine, and we acquired genes that increased our risk for developing obesity today. That in the times of when there was no food around, we had to develop ways to help maintain our fat stores But then when the food became available, suddenly we became fat and diabetic. This theory was loved, but no one could find the thrifty gene. And so it's been largely discarded in a lot of the medical societies. But I'm going to show you today that this thrifty gene hypothesis is true, and I'm going to also show you that it's extremely relevant to this population, and particularly to Pacific Islanders. So what's interesting is that humans are at increased risk for gout because of a mutation that occurred in a gene called uricase. And there's this enzyme in our liver that degrades uric acid. So most mammals have low uric acid, but humans have elevated uric acid. And that mutation occurred in this, 15 million years ago, and it occurred at a time in the Miocene, and it affected all the great apes and humans. And there was a parallel mutation that occurred at the same time in the lesser apes. So all apes and humans have this mutation, so we all have higher uric acid levels. So I became interested in why these two parallel mutations occurred in the Miocene. So I started reading all the literature I could, and I discovered, or I didn't discover, but I found in my readings that that the Miocene was the period where the first apes appeared in Africa. And these were large tailless apes with big heads, and it was a major advancement in evolution. And it was a very warm weather during that time. It was tropical. These apes lived in tropical rainforests, and they ate fruit, and they were basically lived in the trees, and they ate fruit, and their primary food was, guess what? Fructose. That was what they lived on. 90% of their food was from fructose. They did eat some other things that provided other nutrients. And then global cooling occurred. As a friend of mine wrote, a cold wind blew in the tropical Eden because the apes had increased to up to 20 species. It was an idyllic time for these animals. But as global cooling began because of tectonic plate movement, volcanic activity in the Rift Valley, dust went up in the air, it started cooling, polar ice caps began to form, and the water levels began to recede, and Africa, which had been an island, suddenly a land bridge occurred between Africa and Europe, and apes migrated into Europe just like anteaters and elephants and rhinos, and it was a big time where all this migration occurred. Originally, these apes were living in Turkey and in Europe and in Asia. The tropical rainforests continued to be present. They continued to eat fruit year-round. But as global cooling continued, in the rainforests in Europe began to decrease in size, and deciduous forests began, and suddenly these apes started to starve. And actually, if you take, this is from my friend Peter Andrews, who, so I do have to tell you that, when I was reading this, I said, I have to find a world expert on this. And the world expert on the Miocene apes is a guy named Peter Andrews. He trained with Richard Leakey. He's the curator, or was, of the Museum of Natural History in London. And I flew to London to meet with him. And he took me into his back room, and he showed me these teeth to point out that these were 9 different Kenopithecus that all starved to death, or basically they all met a bad ending. But they have these rings. You see these little rings like tree rings? That's actually seasonal starvation. And he pointed out and has written that these apes, as there was a decrease in rainforests, and they couldn't eat fruit year-round, and there was a period of time, the winter season, where they actually started to starve, and they actually became extinct in Europe. But he wrote in a paper, and big in big papers in Science and so forth, they looked at the fossil record and they realized that we are all descendants of that European ape, that some of those European apes made it back to Africa. So just like there's the out of Africa story, there's actually a back to Africa story, and it occurred before modern humans radiated out of Africa, but we actually went out and came back. So our ancestors lived in Europe, and by the fossil record, we actually returned to Africa. And as he explained to me, that as he pointed out, that it was that when the global cooling occurred, there was no benefit, there was no survival benefit to a uricase mutation in Africa, because even though there was global cooling, the rainforest stayed year-round there. They retracted but never went away. But in Europe, where the rainforest disappeared, there would have been a need for an animal to find a better way to store fat. And so remember we talked about how fructose produces uric acid, and remember we told you how uric acid increases fat. So if fructose helps increase fat stores, and as the winters begin and they can't get fruit, the fat stores become depleted, they starve to death. But if there is a mutation that occurred that would increase the uric acid, could that have increased the fat stores, allowing them to survive? So how would a uricase mutation increase fat stores? Well, remember I told you that in animals, you have to give large doses of fructose to cause metabolic syndrome. In fact, you pointed out that that's one of the arguments against animal studies. But if you inhibit uricase in a rat, then suddenly you don't have to give much sugar to induce metabolic syndrome. This is a paper in which we inhibited uricase. So we humanized the animal. And when we did that, suddenly low doses of sugar cause metabolic syndrome, fatty liver, hypertension. It's pretty remarkable. More recently, in a paper that came out in PNAS, the Proceedings of the National Academy this week, we collaborated with a guy named Uric, who actually was able to resurrect these extinct genes. He worked with Steve Benner, the first one to ever resurrect any extinct gene. It's like Jurassic Park. This is the guy, okay? Not only does he resurrect extinct genes, Benner was the first one to make artificial DNA that replicates, and he's the one who's doing those incredible studies on Mars looking for life. He's way out there. He's had like 30 Nature papers. Anyway, he resurrected the uricase for us, and when we put the Kenyapithecus uricase into liver cells, we blocked the effects of fructose to increase fat. So the loss of this enzyme did have a major effect, and we, as a consequence, we are all sensitive to sugar. Everyone in this room is sensitive to the effects of sugar compared to a mouse or a rat or some other animal. So now we have these mutations, and when this uricase mutation occurred, it raised the uric acid from about 1 to about 3. And I actually went to the San Diego Zoo and I measured uric acid in gorillas and animals that also lost uricase. And I also studied the Yanomami Indian, the same Indian that James Neal studied. In fact, it was from his expedition. I got the old serums, and these Indians that are on native diets have very low uric acid. But with Western diet and with sugar, the uric acid levels go up. And with that goes the increased frequency of obesity, diabetes. Uric acid levels have increased over the last century dramatically. The average uric acid went from 3.5 in 1920 to 6.5 in 1980, and they keep changing the rules of what's elevated. And how is this related to here? Well, you look at the top 10 countries, the top fattest countries in the world, 8 of them are from the Pacific Islands. But guess what? Pacific Islanders were discovered to have higher uric acid levels even under native diets. And there's now studies showing that Maori, 2,000 or 1,000 years ago, were developing gout. So these brave people who made it all the way across, left Africa, made it all the way out to the islands, they picked up survival genes that help them survive. They will live. When the Holocaust comes, they are going to continue to live. They are the great survivors, but they are also now the people who are the most sensitive to sugar in our prediction. So, in conclusion, Miocene apes in Europe were undergoing seasonal starvation when a mutation occurred in uricase. The increase in uric acid increased the ability of the apes to increase fat in response to fructose and was a survival advantage. Fructose was a savior, not a toxin back then. It was a savior. But while there was initially a survival mechanism, The discovery of sugarcane, the rise of fructose-containing sugars, is associated with the pandemic. Now we're eating 3, 4, 5 times as much sugar as we should be eating, and this may be directly relevant to the Pacific Islanders, who have even a higher uric acid than most other humans, and that may be the reason we have so much obesity and diabetes here. Thank you.